Perfect. All right. Good afternoon, everyone. It is my pleasure to introduce Gino Carnevale to present his review to us. Gino's from Indiana, which is where he completed his bachelor's degree in exercise science and pre-med preparation from Ball State University. After graduating, Gino traveled through Argentina and stumbled upon an opportunity to work a harvest there. Evidently, he really enjoyed his experience because it led him to the Venn department here at UC Davis. Last harvest, Gino learned about organic and biodynamic practices as the viticultural assistant at Robert Sinsky Vineyards over in Napa. This coming harvest, Gino is heading back to Napa to work in the cellar at Spotswood. In the future, Gino plans to build a skill set and work harvests throughout the northern and southern hemispheres before eventually working for a producer on the West Coast. Gino is really knowledgeable and passionate about viticulture, and I'm very excited to hear his presentation about breeding phylloxera resistance in grapevines. So let's now give our full attention to Gino.
All right, thank you so much, Andre. And thank you everybody for being here today to listen to me talk about some recent advances in our understanding of phylloxera and in resistance to phylloxera in grapevines. Switch slides. Oh, there we go. So I just want to start off with a quick outline of what I'll be talking about today. Start off with a background on phylloxera, and then I'm going to talk a little bit about some advances in our understanding of the insect and of its interaction with the grapevine. And then I'm going to talk a little bit about the advances in our understanding of resistant vines. And then I want to end with a little perspective feature on this research area and where I suggest we go from here. So a little background on phylloxera. We know it's this bug that showed up in Europe in the mid-19th century, and it destroyed a majority of vineyard plantings over a couple decades. These were all own-rooted vinifera, which it likes to feed on. They quickly realized that the bug was transported from North America along with the native grape species that they brought from there. And they found that these species, when they planted them, showed resistance to phylloxera. So they planted them and they grafted on vinifera scions, and they saw a major reduction in vine decline. It's pretty remarkable that the mechanisms that were present in those rootstock material are the same mechanisms that we see today in our current rootstock selections. We have developed more rootstocks for other reasons, but more or less the mechanisms and the genes that we pass forward are the same. And phylloxera has not really been able to overcome that to a great extent. But it does beg the question of for how long and how well will this resistance hold up to phylloxera as it continues to spread and diversify and adapt to different environments in different wine growing regions around the world. So what is phylloxera? We classify it as an aphid-like insect, meaning that it feeds by sucking sap from a plant. It reproduces rapidly and often reproduces without mating through asexual pathways. It forms large colonies and can be devastating to grapevines it feeds on if infectious microbes are able to enter the feeding wounds where it is feeding. We know that its life cycle can vary and is somewhat complex. We have an understanding that there are populations that cycle through the soil and feed on the roots. Other populations cycle through the leaves, feeding on the leaves. And there's some change going on or movement between the root populations and the leaf populations. We know that they often reproduce asexually through a process called parthenogenesis, producing more or less clones of the mother insect. But we know that there are other cases where they reproduce sexually, resulting in genetic recombination, which can make understanding the genetic structure of any given population a little bit more difficult to understand. We know they're highly adaptable because of this. And I also mentioned that they're hard to kill, which is why we use rootstocks instead of finding another sort of insecticide product, because they like to dwell in clay soils where it's difficult to effectively inject any sort of insecticide product that really reduces the numbers. And on top of that, they reproduce so rapidly, so many generations in a growing season, that even if there were to be a good product to limit the populations, they would bounce back within a given growing season. So going off of that, something we've learned recently about the interaction is that populations within any region or site do not show much genetic structure. And they theorize, the researchers theorize that this has to do with the complex life cycle with sometimes sexual recombination or reproduction and sometimes this asexual reproduction. So you can have a population that gets introduced onto a vine and continues to reproduce asexually with no sort of genetic changes happening. And then there would be a period of sexual reproduction where you have genetic recombination happening. And then you see a different strain develop on the given vine or within the given site. And you see that so many times throughout a given period that it develops a large diversity of populations within a given site, within a given region. Makes it a little bit more difficult to really understand the population dynamics of a given place. Another thing that they found recently in this study by Forneck over in Austria and Switzerland is that there may be populations that are adapting to the grafted vine system. So we knew that there were Phylloxera populations that prefer American species rootstocks. Even though these rootstocks don't decline, these populations like to feed on those roots. And we know there are populations that like to feed on leaves, mostly on American species leaves, whereas for a time we've seen some sort of resistance of vinifera leaves to this leaf feeding form. This is the first time reporting populations moving from the roots of an American species and as the rootstock onto the leaves of the vinifera scion and feeding on that and forming these galls that you see here in this picture. This isn't exactly a major issue because we know that the leaf feeding form does not cause the same sort of vine decline that you see with the root feeding form. But it is interesting and it's helping researchers understand a little bit more about how Phylloxera is adapting to these different conditions that it's in. Another thing that they've learned recently, looking at the saliva of Phylloxera, is that it releases these effector of the vine, inducing the formation of these galls that you saw in the last picture. So this study here is basically they were doing a profile on the different proteins, and the Venn diagram that you see is basically the results of a blast sequencing that they did of all these proteins they collected from Phylloxera samples. And as you see, these proteins are pretty homologous across different phytophagous insects, which for the researcher can be beneficial in helping us sort of understand more about the mechanism of how these proteins are impacting the induction of genes that are expressing these galls. So maybe researchers are able to now look at other insects that also have these proteins and look at the mechanism from a broader perspective, which could be interesting and productive in that way. Yeah, let's go through. So another thing that they've been doing recently is looking at the different biotypes and trying to characterize the biotypes in different regions around the world. What I mean by biotypes, around 1980, after the collapse of the AXR1 rootstock, they determined that there was basically Phylloxera was adapting and preferring whatever material it was feeding on and specializing on that. And so today we now have 7 different biotypes characterized by the material that they prefer to feed on. The big ones in California are biotype A and B. A being the biotype that prefers to feed on vinifera roots, where B is this biotype that showed up and started to feed on the AXR1, which is a crossing of an American species, rupestris, with vinifera. And now there are other biotypes. As I said, there are 7 of them now that prefer to feed in other ways on different rootstock material. So the characterization of the strain populations in the different regions of the world is being done not only to help wine growers in that region understand what biotypes they have and what rootstock material they should be using to deal with that population. It's also helping researchers understand how populations are moving around the world and how different regions may be introducing Phloxera populations into a new region. So for example, in this figure we have from a paper a couple of years ago done in Argentina, looking at the populations and regions there. They found that some of the populations were more genetically similar to the biotypes that we find in California and the populations we have here, whereas other populations they were looking at were more similar to the biotypes and populations they have in Europe. So that's helping them understand how maybe some of the populations were introduced from rootstock material that was transported from California, which can help producers and researchers understand maybe a little bit more about how phylloxera is being transported and developing better systems to prevent that from happening in future transportation of rootstock material. So some things that we've learned about resistant vines. This figure on the right is looking at a paper that was done classifying the first gene for resistance to phylloxera in a crossing of Vitis cinerea with Riparia, and that was called Borner, which they crossed with Vinifera, Riesling strain. And because they knew Riesling and Riparia were not highly resistant to this population, they concluded that the gene that they found correlating to the expression of resistance came from the Vitis cinerea parentage. And then in the more recent study a couple of years ago by Smith et al., they found with a similar crossing of straight Vitis cinerea with Riesling, a new gene on chromosome 14, which they classified as RDV2, a second gene for resistance to phylloxera. But I think it's kind of interesting. I take this results in the more recent study with a grain of salt because they conclude that this new gene RDV2 comes from the Vitis cinerea parentage, but they don't find in their results the RDV1 that they concluded was from the Vitis cinerea parentage. So I was kind of confused about whether this new gene is actually either coming from Vitis cinerea or if it's even actually separate from RDV1. I think there needs to be more research and following up on that. But it is interesting to know if there are multiple genes for resistance. Another thing that we've learned is that there are now two separate hybrids with different parentage that are showing a resistance gene on chromosome 14. So the same results from Smith et al. A couple of years ago, looking at chromosome 14 with that Vitis cinerea by Riesling. And then the figure on the right, this was a study looking at samples that had a parentage of a combination of 6 different Vitis species, Vinifera, Riparia, Rupestris, Labrusca, Estivalis, and Berlandieri, not including cinerea. So the only problem with the results here is that it was just a quantitative trait that they identified without doing any sort of simple sequence repeats to really hone in on the genome, on the genetic sequencing for that quantitative trait. So they really need to follow up. Somebody needs to follow up on the results of this other paper to confirm whether this is also RDV2 that they're looking at, or if it's another gene for resistance to phylloxera, maybe a potential RDV3. So a few other things about resistant vines that we've come to understand. Crossing with Vinifera is going to require more research and potential genetic modification if we want to produce rootstock material that has Vinifera parentage. After AXR1 and the collapse of that rootstock material, researchers learned that they developed a theory that the gene for resistance, the allele for it is homozygous recessive, and that potentially Vinifera has a dominant allele. So if you have any crossing with Vinifera, you're going to get a loss of that of that resistance being expressed. So if we want to use that vinifera in any future crossings, we might need to develop a way to remove that dominant allele. In addition to that, durability of resistance is a really important factor to consider, and the best pathway we have right now to developing rootstock material with a more durable resistance is to develop a gene pyramid. So going back to what I was talking about with the identification of the genes RDV1 and RDV2 for resistance, we now have technology, marker-assisted breeding, that can allow us to really follow the genes through a breeding program and develop material that contains multiple genes for resistance. And if you have plant material that has this gene pyramid with multiple genes of resistance, then it will take a longer time for Phloxera populations to adapt and overcome all those different expressions of resistance within a given material. And so some suggestions for what I think we should do next. We need to continue to characterize the diversity of biotypes around the world for the reasons I explained earlier, helping researchers understand how populations are moving and adapting and diversifying. And in addition, helping wine growers understand the biotypes that they need to be thinking about and dealing with in their plantings. I also think that we should investigate the effects of soil microbiota diversity and overall vine health on resistance to phylloxera and other pests and diseases. There is some anecdotal evidence that wine growers using organic practices, planting own-rooted vinifera vines on soils that have previously had issues with phylloxera are not declining to phylloxera, and they theorize that it has to do with the healthy soil microbiome that they've developed through their practices. And as I remind you, vines don't directly decline because of phylloxera feeding. It's because of the wounds that they form are then infected by infectious pathogens. So if there aren't very many infectious pathogens within the soil, then potentially the phylloxera population feeding on the roots would not be as big of an issue. So that needs more research. And in addition to that, we need to maintain a diversity of rootstock material in our vineyards. We know that the resistance that we have in our rootstock material has held up over a long period of time. But we know that phylloxera continues to evolve, and it can develop systems to overcome the mechanisms that these materials have for resisting phylloxera. And so if you only have one rootstock material type in your vineyard, and a phylloxera population is able to overcome that mechanism, then you're going to see your entire vineyard site start to decline more rapidly than if you had a diversity of rootstock material with different mechanisms and different pathways to resist the phylloxera population. And that is all I have today. I want to make some quick acknowledgements. I'd like to thank Andy Walker and Dario Cantu for their advising help with this presentation and my literature review. I'd like to thank my former boss, Carrie Flores at Robert Sinsky, for really developing my passion for viticulture and just the vineyard life and trying to understand more about how these vines operate. And I want to thank my cohort. I'm pretty bummed that we can't all celebrate this last quarter together, but I look forward to when this quarantine's over and we can all get back together. And finally, I want to thank the different groups that helped me fund my education here at Davis: the Wine Spectator Scholarship, the Wine Institute Scholarship, the UC Davis Departments of History and B&E that gave me positions as a TA, and then the union for the TAs that has negotiated a way for me to be able to turn my TA position into funding my education. So I want to thank all these people, and then that's it. Thank you.
All right, great presentation, Gino. So this is the only presentation we have today, so we have plenty of time for questions. So if you have a question for Gino, you can either directly unmute your microphone and ask, or you can raise your hand on the sidebar and we can moderate questions that way. All right, it looks like Malenka has a question.
Yeah, hi Gino, great presentation.
Thank
Um,
you.
I was really interested in the sexual reproduction of phylloxera and how that's, creating genetic diversity for it to overcome resistant rootstocks. Did you find a lot of research in when sexual reproduction happens, or how it happens, or how it might be blocked? I know there's some pheromones that block other insects' reproduction. I don't know if you've found anything like that.
The most recent research has really just been confirming that there is sort of sexual reproduction happening, but that's kind of just the first step in determining how that can be stopped. So maybe there's current research going on into that, but I didn't really find anything into people looking into the specifics of how exactly it's happening and what pathways can be inhibited, I guess.
Cool, thanks. So,
we have a question from Spencer.
Hey, can you hear me?
Yeah.
Cool. I was just curious. A lot of my neighbors have some really old vines, and I was just curious if you looked into much of, if you're on vinifera rootstock and actually trying to control phylloxera, because I was just curious. There's some like 150-year-old vineyards around here, and they're still harvesting today, and they seem to be spraying some type of insecticide to control the phylloxera. And I was just curious if you looked much into it. In your presentation, you're talking about how it seems like most of the insecticides aren't really that effective, but I just was curious if you looked much into that.
Yeah, I didn't look too much into the specifics of what products are really being effective or not. I know that they've developed a product that works pretty well on the leaves for reducing the leaf feeding form populations. But I just generally found that people that have been trying to develop products for soil are having issues because really the populations like the clay, and it's hard to inject anything into a clay soil and have it work. So.
And then can I ask another question off that?
Yeah.
I was just curious. So if you see a lot of galls on your leaves, that's not indicative of having a huge population in your soil. There's no correlation there, or?
I wouldn't say that. I know that if you have a large leaf feeding population, that at least from what we're seeing now, that there is migration between the leaf and the root, and that if you have generally a population of phylloxera in your vineyard, it can be moving into the soil or onto the leaves. But I guess it would just take more sampling to confirm whether you're really seeing that wherever you're seeing the galls on the leaves.
Cool, thanks. Nice, great presentation.
Thank you.
Guillermo has a question for Gino.
Go ahead. No, maybe his microphone's off.
Yeah.
Guillermo, I can't hear you. Maybe if you type it, I don't know. Yeah, that works.
All right, well, Guillermo figures out his mic. Kara has a question.
Go ahead.
Can you hear me?
Yeah.
So just a quick question. Has there been any research, especially now that there's more research done on the leaf feeding form, whether phylloxera is able to transmit virus, disease?
That's interesting. I didn't find too much research into phylloxera as a vector. We know that its feeding wounds are basically opening up the roots to any sort of pathogen that's in the soil. But I don't know if there's anybody really looking into specifically phylloxera as a vector transmitting any certain diseases.
Okay. But they haven't, and they know that the root feeding form opens it up for disease, but they aren't doing like a disease screen on those phylloxera to see if they're carrying any viral vectors or viruses.
Not from what I found. No.
Okay, interesting. Thank you.
Yeah.
Guillermo says, recently Phylloxera woke up in Washington State, where most of the vineyards are planted without rootstock. Have you heard about any research that studies potential triggers for that?
Potential triggers for a population to just start to take over. You know, I didn't find anything on that either. It could be just a changing in the environment that allows the populations to really blow up. Maybe it's getting warmer. There are people looking into the effect that, you know, a warming climate is going to have on the spread of phylloxera. But so that, I guess that could be an interesting thing to look into. But I don't know of any other research about that. No. Thank you.
All right, are there any more questions for Gino? All right, looks like there are no more questions. Good job, Gino, good presentation.
Yeah, thank you, Andre. Thank you. Thanks, everybody.
Good job,